I'm not going to listen to anybody. So I am planning to do your mine biology, and I'm gonna start from the beginning. So fundamental of cells. Cells are in living and living cells are living cells. Cells are basically living. And first is your unicellular, second is multicellular. Division in liver and multicellular. Unicellular example are bacteria, amoeba, yeast. These are the unicellular and multicellular. Basically us, but in this cell have division of work. So like heart would pump the blood with oxygen, and yeah, that like intestine would really would absorb the nutrients. Then 1665, Robert Hooke, he discovered the structure of cell, and you could say cell as he shown the structured as a honeycomb stacked. Honeycomb type thing. Yeah. And then 1674, uh, Antelo Antino Leeuwenhoek, and Leeuwenhoek, he found, so 1674 and 1550, sorry, 1665 and 1665, so 1665 Robert Hooke, and 1674, so 1665 and then 74, so like 9 or something, yes. And so 1665 and 1674. Antino Leeuwenhoek, he took a pond sample. He took water from a pond, and he got the living cell, free living cell in that bacteria of sample. Next is cell theory, in which if 1858 Shendon and Swan SS, all living cells are made up of cell and are Benodomental of unit of life. 1830, 1838, 39, Shendon and so on. 38. 1838 and 39. Shendon.
Live चलेगा भाई. Live, my brother's live. Hello guys. If you like this video, please like, share, and subscribe. He's a really intelligent guy. If you can learn from him.
Shendon and Swan, they discovered the all the living cells are made up of your all the living cells are made up of bacteria, not bacteria, like all the living cell are known to be basic unit of life. So SS discovered basic unit of life. Not discovered, they just tell that cell is the one. Next is your 1855 Rudolf Virchow. All new cell will arrive from pre-existing cell. Rudolf Virchow.
See, Vic.
राजा बंद करे Rudolf या Rudolf Rudolf wolf like wolf is like this, right? Rudolf Wolf Rudolf Vashko Vescho Rudolf Vescho. He said that all the living cells are made up of pre-existing cells. Rudolf Welchow. So Wolf and Cho. Cho is the fighter in MMLB. सो चो एंड वुल्फ ने डिस्कवर्ड करा कि एक जो सेल है अगर चो उसको मारेगा तो वुल्फ उसको रिबॉर्न करेगा और वो सेम सी होगा. ओके. Rudolf Virchow in 1885 or something. 1855, sorry. 1855. 1855. 55. Five wolf, five chos attacking each other and making a new cell. Smallest cell is oval is microplasm galliptism, and human is sperm cell and female granule. Female में granule cell. Smallest cell is microplasm gallipsiptum. Microplasm gallipsiptum. Microplasm gallipsiptum. Its size is 0.2 till 0.3 micrometer. Microplasm gallipicum. So I did Robot Hook as of 1665. Then I did that one guy with Pond one, which was Antino. Leeuwenhoek was the border one. It was 1674, and Robert Hooke was 1665, and Leeuwenhoek was 1674. Then I did some two brother SS, and they were in 85, 55 or something. Two brothers or 38, 39. So Shandon and Swarden, There's a basic structure of life. Then I did your 55 one, which is wolf and your cho. So Rudolf and Vescho, living cell and have pre-existing cell. Like the cells are made from the pre-existing cells. Cell membrane. Cell membrane is an outer layer of cell, extra rigid and hard, dead layer. Okay, plants only have them. In fungi, it is cactin, and bacteria, it is peptidoglycan. So Robert Hooke is in your 1665 discovered the basic structure of the cell. Then Leeuwenhoek is in your 1616 70 something 1674. And Leeuwenhoek is in 1674. Then there is Schwann and what is this? Schwenden and Schwann. So Schwenden and Schwann in 1938 and 39. Then there is your Wolf and Cho. What is his full name? Rudolf Wenschow. It is in 1855. Okay. Then cell membrane I did. It is an it's it's an outer layer which provide extra rigidity. And then this cell membrane, the plants have a normal cell membrane of cellulose, so cell is of like basically cellulose. Then fungi fungi have a cell membrane of your pectin, I think so. Sorry, chitin. Then bacteria has of peptidoglycan. Bacteria cell membrane is of peptidoglycan. 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 So plants have a cell wall of cellulose. Fungi have it of chitin. Then bacteria have it of peptidoglycan, right? Peptidoglycan. Peptidoglycan. and this whole field is called your pitch, and above pitch it is called lamelle, and this thing is called midrib, mid cage or something, mid backbone, no, midrib, I think it's called midrib, and this holds it. It's also called something, I forgot. Yeah, I've had in my different notes. So the outer layer which is protecting it is a cell wall, and cell wall is only found in your plants as of cellulose, fungi, as of fungi. Inside of fungi, this thing is found as chitin. Then in front of in bacteria, pepito pepito glycan, whatever. No, pepito glycan. Put it here. Okay. Yeah, cell membrane is semi-permeable. It doesn't allow water to go. It allows water to go outside, but doesn't allow water to come inside from the leaves. I might be wrong. It is something else. I don't know. Plasma membrane. It is defined as the cell region, living, flexible, uh, secretory permeable, lipids, protein, fats, pore size, uh, contraction of higher to lower osmosis and lower to higher reverse osmosis. So water and liquid. Okay. Then there is protoplasm. Protoplasm is Projiki term in 1839. So with double S brother, Schwann and what is his name? Shenden and Schwann. Concentrated quantities to lower osmosis, diffusion of water and liquid, lower to higher reverse osmosis. So, Plasma membrane, flexible, rigid. Pores are there. Opening of pores is called opening and closing of pore from higher to lower. It is called osmosis, and lower to higher is called reverse osmosis. Then medium and plasma membrane, hypotonic solution. Okay, isotonic solution. Okay, I got it. This one though I know. Here it is. Medium and plasma membrane. In this thing, there are three things. First is hypertonic solution. So in this, the quantity of your solute, which is your whatever the acid or the main juice you could say, solute is higher, and the solvent, which is water, it is lower. And in this, if this is the cell, then it will shrink because it doesn't have water like resin. Now it is isotonic isotonic solution. In this, the solute and solvent are equal. So in this, the shape of the cell inside of it will remain same, would be perfect. Now hypertonic, isotonic, and then now it is hy- hypotonic, hypertonic and hypotonic, hypertonic, hypotonic solution. Hypertonic, hypotonic, water is more, so solute is less and solvent is more, and the inside cell would bulge like in animal cell. It would swell up, and in plant cell increase the turgid presence. Okay, now there is nucleus. Nucleus is the brain of the cell, and nucleus is basically brown, you could assume. So 1831, before the 38 and 39, Schwanden and Schwann brothers. 1831, the Brown, what Brown is Robert Brown. Robert Brown discovered nucleus. A nuclear membrane start with membrane. Nuclear membrane, what the heck? Nucleus. So this is nucleus membrane, and it is a dou- double membrane, and this thing is fluid. Double membrane fluid. Robert Brown, the 1831. Nucleoplasm, fluid nucleus related. So nucleus plasm. Then the thread-like structure is called chromatin, and chromatin opens up and arrange linear chromoplasm chromosome DNA plus protein. Thread-like structure is made up of chromatin, and it opens up and arrange linear, and this become chromosome is came from your thread-like structure in the nucleus, which is chromatin, then chromosome, and in this there is DNA and your protein, 23 chromosomes and all that. And nucleus. Then nucleus. Nucleus is made up of your RNA plus ribosomes, which ribosomes can be rough endoplasmic reticulum and smooth endoplasmic reticulum. So smooth hair doesn't have ribosome, which ribosomes are your protein factory. Tienes dioxinazole. So then now short full form of DNA is deoxyribonucleic acid. Dioxy dioxyrib nucleic acid, and it is the main purpose is genetic material. Genes found in nucleus and mitochondria and plastid. Semi-autonomous organelle. And I just read it, man. I'm not writing this much. RNA are rare and RNA doesn't have uris. So RNA has different things. RU so uracil and DNA have your thymine. So DNA have thymine and RNA have uracil. Everything else is similar. Uracil and DNA have your thymine. Then nucleoid, undefined nucleus region in prokaryotic. Okay, nucleoid is that. Then endoplasmic reticulum, Keith Potter. Harry Potter ने बनाया endoplasmic reticulum. Okay, okay, okay. Endoplasmic reticulum, Harry Potter. Not Harry Potter actually, but Cathal Potter. In forms of tubes and gesture, found near as well as from nucleus. Rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough प्रोटीन सिंथेसिस करते हैं, स्मूथ वाले फैट प्लस लिपिड सिंथेसिस होता है. तो स्मूथ वाले फैट होते हैं, रफ वाले प्रोटीन बनाते हैं. Oh, I don't have ribosome. Oh, okay. Rough endoplasmic reticulum have ribosome, right? Ribosome. Smooth endoplasmic reticulum have don't have ribosome. Okay. Vegetables, liver, smooth endoplasmic reticulum, capillaries, and removal of toxics and subs. Okay. Mitochondria, powerhouse of the cell, fluid, the inner membrane, crystal. Okay. Fluid called matrix. In mitochondria, you are moving in your matrix in the fluid. The cell membrane pores. ATP, energy conversion rate, and AOP. And ATP is conversion of energy. AOP is adenosine diphosphate. Helps in cellular respiration. ATP produced nutrient into energy in cellular respiration. Glucose plus O2, cell mitochondria के beside glucoblaca energy ADP ATP. Golgi apparatus. Brother, I am not doing this much. This looks smaller, but it is a lot.